In this video, we will use OpenCRAVAT to analyze somatic mutations. These mutations are obtained from exome and genome sequencing of 76 untreated metastases from patients with breast, colorectal, endometrial, gastric, lung, melanoma, pancreatic, and prostate cancers. These 15,765 mutations are available as a tab-delimited file at opencravat.org. On run.opencravat.org, upload the tab-delimited variant file and select the HG19 genome version. Next, select the following from the annotations section: cancer hotspots, cancer gene landscapes, CHASMplus, CIVIC, COSMIC, mute panning, and index NCI. Finally, click the Annotate button. It may take a few minutes for OpenCRAVAT to process this data set. Once completed, open the job in the interactive viewer. The Cancer Gene Landscapes annotator identifies oncogenes and tumor suppressor genes described by Vogelstein et al. To find variants within these genes, navigate to the Variants tab and click on the class header under Cancer Gene Landscapes to sort. These variants occur within tumor suppressor genes, or TSGs. Alternatively, the search box can be used to find variants within oncogenes by typing in that search term. The COSMIC annotator includes information from the Catalog of Somatic Mutations in Cancer, a high-resolution resource for exploring the genetics of human cancer. In OpenCRAVAT, the COSMIC annotation shows how often a mutation is observed in different cancer types. For the first mutation in the sample, a Missense mutation in MPL, it has been observed 4 times in skin cancer. The mutation has not been observed in other cancer types in the COSMIC database. Driver Missense mutations. The CHASMplus annotator is a prediction method that finds mutations that are likely cancer drivers. On the Filters tab, click the checkbox next to CHASMplus to apply a filter for values with a p-value of less than 1,000th, and then click Apply Filters. In total, there are 95 mutations in this set that are predicted as drivers with a significant p-value. To see which genes are impacted by these Missense mutations, click over to the Gene tab. Then, click twice on the header of the Number of Coding Variants column to see which genes have the most potential driver mutations in the data set. TP53 is affected by 6 Missense mutations in this sample with significant CHASMplus p-value. The locations of these mutations are shown in the protein diagram on the lower half of the screen. The mute panning column shows that TP53 is mutated frequently in cancer, with 24 different cancer types showing a significant amount of cancer mutations. In contrast, the second entry in the table, KRAS, has 6 mutations but is only seen in 11 cancer types. To see the NCI cancer pathway networks that involve the TP53 gene, scroll down in the widget pane to view the index widget. The drop-down at the top of this widget can be used to select among the networks that contain TP53. In this network, the TP53 gene is shown in red and can be used to find direct interaction partners. Clinically actionable mutations. On the Variant tab, we can use CIVIC to find clinically actionable TP53 mutations. First, type TP53 in the Gene column search box to restrict the Variant tab to variants that impact TP53. 2 of these mutations have annotations in the CIVIC column. Click the plus button to expand the CIVIC section to show all of the CIVIC columns. The first mutation has an actionability score of 22, whereas the second has an actionability score of 0. The first mutation has much stronger evidence to support actionability. This particular TP53 mutation is related to several cancers. To learn more about the mutation's actionability, use the link in the ID column to be directed to the CIVIC website. By integrating these resources, OpenCRAVAT makes it easier to perform in-depth analysis of cancer-related somatic mutations. To learn more about which other cancer annotations are available, go to run.opencravat.org.